So in this video, we will look at enzymes, industrial enzymes. There are a lot of industrial enzymes produced by microorganisms, especially bacteria and fungi, which are widely used in industries with great effect. So the class of enzymes include oxidorubtases, lipases, which act on lipid substrates like phospholipase, esterase, lipases, etc. Say cellulases, another class of enzyme, amylases, which helps in the hydrolysis of starch, cellulases, which is widely used in paper industry, and the pectinases, clarification of juices. But the enzyme in general, industry in general, is being dominated by. So proteases are widely produced by microbial sources. So microbial sources are among the best sources compared to animal and plant sources to be utilized in industries. So when you say proteases, protein can be produced extracellularly or intracellularly by microbial sources. So it can be acidic, it can be alkaline or neutral proteases based upon our utilization in the laboratory or industry. We can use them. I want to screen microbial source that produce protease, which has hemolytic property and as well as keratinolytic property. So where I can use that is my primary aim here is to isolate a strain, produce strain which has keratinase activity as well as hemolytic activity, so that I can use them effectively on slaughterhouse waste or biomedical waste remediation. So this is how I can do that. I isolated them on a variety of sources and plated them on casein agar plate. So based upon the hydrolysis of casein agar, I can isolate the best source. So you can see that the protease hydrolysis in casein agar plate. So now that I have isolated a protease enzyme producing bacteria, a high yielding strain in fact, I want to see whether they perform hemolysis. So I further put them on blood agar so that whether they can actually degrade blood. So now that I found a strain that can degrade blood, heme as well, so that you can see that lysis of blood on the plate. So now that it can also degrade blood or heme protein, whether I have to see whether it can degrade keratin as well, which is usually a very important ingredient of slaughter waste. I found certain strain has a very high keratinase activity as well, along with the hemolytic activity. So now that I have isolated the strain, I want to go further, purify these proteins, and I'll meet you in the next video for further. Thank you.